Okay, students, here we are on slide 12. I've skipped the first 11 slides about how prokaryotes reproduce themselves because that information is pretty easy to understand. What's a little bit harder is how eukaryotic cells grow and what their life cycle is like, or what their cell cycle is like. So I'm going to lecture on this part. We've learned a lot about eukaryotic cells in that they're a whole lot more complex than prokaryotic cells. For one, they're larger, and they also have more genes, more genetic information. And those genes are stored on multiple chromosomes inside of the nucleus. So the process here becomes just a little bit more complex for a eukaryotic cell. So inside a eukaryotic cell, we see the chromosomes. And that's kind of a word that you probably heard before, but you may not have ever had explained to you what exactly is a chromosome. And chromosomes are composed of what we call chromatin. And chromatin is made of really a long DNA molecule and then proteins. So you can kind of think of chromatin as being a spool of thread, where the spool is the protein and the thread is the actual DNA. So in this picture, they do a pretty good job of showing you where the proteins are, right here. We call those histone proteins. And then the DNA is here in these long purple threads. So the function of those proteins is to organize the DNA. So the DNA kind of wraps around those proteins, and it helps to keep the chromosome structure, or it helps to maintain or prevent tangling of that DNA. DNA is very, very long. So before a cell divides, what you'll see inside of the cell is that the chromatin condenses. It becomes very visible underneath a microscope when we add a stain to the cell. So let me just show you kind of what's what here. That's the plasma membrane. And then here, what you see in the center, that's the nucleus. So we'll just draw in the nuclear membrane as well. And those purple strands that you see, that's the chromatin. Normally, if a cell is not preparing to divide, you wouldn't see the chromatin condensed like that in the nucleus. It would be more spread out. But just before a cell begins to divide, that chromatin becomes really condensed and it becomes visible beneath a microscope. Now think about if you wanted to divide, or you wanted to make a copy of yourself. You'd have to copy all of your contents. And the same is true then for these eukaryotic cells. Before a cell begins to divide, it has to duplicate all of its chromosomes. And not just all of its chromosomes, but all of the other organelles on the inside. And so when a cell duplicates its chromosomes, it makes a copy. So these copies now we call sister chromatids. Think of them as like twin sisters, identical twin sisters. And they're joined together at a structure called the centromere. I'll show you a picture of that in a second. And then when the cell divides, those copies, those sister chromatids, they separate from one another. One chromatid will go to one cell and one chromatid will go to another cell. So when they separate, that's when we call them chromosomes. So let's have a look at this. So here's just one individual chromosome prior to replication, and then the book shows you what it would look like here. The actual DNA would look like, we call it a DNA helix because of its spiral shape. So once those chromosomes duplicate, you essentially have what we call sister chromatids, identical twin sisters. The chromosome has made an identical copy of itself. And then when a cell divides, when one cell is going to become two cells, those sister chromatids are going to have to separate. So that's what you see here. Now you've got those sister chromatids separated, and each one of those chromosomes will now go to a separate cell. Before we learn about the process of cell division, the process that we call mitosis, there are actually two ways in which cells divide. We're only right now concentrating on the process of mitosis, and mitosis is the process where cells just make an identical copy of themselves. So they're really just making clones of themselves. In another lecture, we'll cover the other type of cell division called meiosis. And this is a little bit more complex. Now, cells aren't going to be making identical copies of themselves. They're going to make slightly different copies of them, not copies. They're going to make slightly different versions of themselves. But for right now, we're just going to concentrate on mitosis. So the picture that you're looking at here is a picture of what we call the cell cycle. You can kind of think of this as like the cell's life cycle or your life cycle, all of the things that you do within the course of your life. So the cell cycle is broken up into two main phases. So we have what we call interphase and what we call the mitotic phase. So in interphase, the cell is kind of doing its daily things, its daily activities. And in the mitotic phase, the cell is preparing to divide and make a copy of itself. We'll go over each one of these sub-phases in a little bit. So within interphase, there's what we call G1. Within interphase, there's what we call the S phase or synthesis. And within interphase, there's what we call G2 or the second gap. All of this is happening prior to mitosis. Okay, so this is kind of what this text right here is just kind of explaining what the previous slide was showing you in a picture. And I'll just kind of reiterate here. During interphase, what the cell is doing is duplicating its contents. It's duplicating the mitochondria, it's duplicating the endoplasmic reticulum, the ribosomes, all of the things that cell is going to need in order to make a copy of itself. In addition to that, it's duplicating its genetic information. It's duplicating its chromosomes. So if we look at it just in interphase, we're not in mitosis yet, we're looking at interphase. The first phase of interphase, the cell is just growing, it's doing its daily thing. But also it's going to increase its cytoplasm, or it's going to increase all of the contents. That includes all of the organelles, plus the fluid that's in the cell also. After that happens, the cell then enters the S phase, and that's where duplication of chromosomes comes in. And then in G2, the very last phase of interphase, the cell's going to continue to grow some more, but more importantly, it's going to prepare for division. It's going to pretty much prepare to divide in half and make a new copy. And that dividing in half, the complete separation of all of the chromosomes and all of the organelles into two separate cells, that's what we call mitosis. If we look a little bit more closely at mitosis, or the mitotic phase rather, mitosis is actually the division of the nucleus. And remember, inside the nucleus is where we find chromosomes, the DNA, that genetic information, your genetic instruction booklet. So in the mitotic phase, mitosis occurs first. We divide up the chromosomes. And then second, what happens is division of the cytoplasm. So the cytoplasm, remember, is organelles as well as the cytosol, the fluid inside of the cell. Okay, so let's look a little bit more closely just at mitosis, the process through which the nucleus is dividing, or the chromosomes are going to separate. So let me just name the phases of mitosis for you. The first one is called prophase. Pro means actually first. So this is the first phase of mitosis. Your book also explains a phase called prometaphase. I'm not going to cover that here with you, but I wanted you to see the term and kind of understand it in case you read about that in your book. You can kind of ignore it for testing purposes. It's just an intermediate phase between prophase and metaphase. Okay, so there's prophase, that occurs first. Second is what we call metaphase. Third is what we call anaphase. And fourth is what we call telophase. In the Amoeba Sisters video, they ask you to remember PMAT, so you can remember the order of these phases of mitosis. The second subphase of mitosis is what we call cytokinesis, and a lot of times that kind of is occurring at the same time telophase is. But remember, cytokinesis is dividing up everything else. So everything else then, of course, would include all the organelles and the fluid inside of the cell also. So I'm going to show you pictures of this in a second, and the Amoeba Sisters video and several tutorials out there are going to go through this process with you. So if you don't understand it completely after this lecture, feel free to go ahead and look at any tutorials that you find helpful. Okay, so remember that mitosis is division of the chromosomes, or dividing up the contents of the nucleus. Those chromosomes are in the nucleus, right? So before those chromosomes can be separated from one another, there's a structure inside of the cell that develops. And this structure is called the mitotic spindle. And you can kind of think of the mitotic spindle as being like a Velcro glove, where it's going to attach to the chromosomes and pull those chromosomes away from one another. So that mitotic spindle is composed of proteins, what we call microtubules. And those microtubules are created or produced by organelles inside of the cell called centrosomes. I'll show you a picture here in a second. So let's have a look at this. We'll see the centrioles here in a second. So remember that what the cell is spending most of its time in is interphase. This is not part of mitosis. Remember, this is that other part of the cell cycle. Let's just look at interphase first. Let's look at some of the structures that are present. So in interphase, the cytoplasmic contents double. So that means everything inside of the cell doubles. And then more importantly, let's look at the centrosomes. These wouldn't normally be present. They're only present when a cell is getting ready to divide. So here are the centrosomes. Oops, let's get this. There we go. Here are the centrosomes. And inside of those centrosomes, you can see the centrioles, and those are paired. So remember that during interphase, the chromosomes are going to duplicate, and that occurs in the synthesis phase of interphase. So here you can see the cell is preparing to divide, and look at how visible the chromatin is. Another thing I want you to focus on is that you can really clearly see that nuclear membrane, or what we call the nuclear envelope. That's still present, but that's going to disappear. That's this container around the chromosomes but those chromosomes aren't going to be able to separate from one another unless that nuclear envelope disappears. So that's something to watch out for in the next couple of slides. Now let's move into mitosis. And I'm not sure why the S is missing up there in mitosis, but I'll add it in. Remember, PMAT. So the first step in mitosis is what we call prophase. And in prophase, what you can see are the sister chromatids. Remember, those are identical copies of chromosomes, so those are very visible. And then also what you see are the centrosomes, here and here. So there's a big difference now between what those centrosomes look like at the end of interphase and what they look like now. Look at how these long microtubules have extended. And also these centrosomes, they're moving to opposite sides of the cell. You'll see that also in the next picture, or in the next slide. Let me change pens here real quick. So these microtubules right here have gotten a lot longer. Think of those as the fingers on your Velcro glove. They're extending and getting longer and longer. So this is what prophase looks like. The next phase is metaphase. Now look at the difference here. One of the major things you should recognize is that there's no more nuclear envelope. That's disappeared. The idea here with mitosis, remember, is that we want to separate chromosomes into two different cells. That can't happen if there's an envelope or a structure enclosing those chromosomes. Another thing I want you to recognize is look at the positioning of the centrosomes. They're now on completely opposite ends of the cell. Sometimes those ends of the cell are referred to as poles. Think of that like the North Pole and the South Pole. And then let's have a look. Look how much longer those microtubules have gotten. And another thing to look for or to recognize is that some of those microtubules have attached themselves to a point on the sister chromatids that we call kinetochores. So that's just a point of attachment for those microtubules. So now we've got the microtubules attached to those kinetochores. And what you'll see in the next phase is that those microtubules are going to shorten. And because they're attached to the kinetochores, they're attached to the chromosomes at the kinetochore, when those microtubules shorten, they're going to pull those chromosomes away from one another towards the poles. So then that brings us to this next phase called anaphase. And what you'll see again are the chromosomes are starting to pull away from one another. So here's one chromosome, here's another. Here are the microtubules. They're starting to shorten, and that pulls those chromosomes away from one another. Another thing to keep your eye on is that the cell is elongating. And some of these microtubules right here that are not connected to the kinetochores at the chromosomes, they're pushing the two poles away from one another. Because ultimately our goal here is to split that cell in half. So the cell's getting a little bit longer. And then that brings us to telophase. The cell is still getting a little bit longer. And one of the things I want to draw your attention to also is, the nuclear envelope or the membrane around the nucleus is now coming back. It's going to reform. Another thing to look out for is this. That's what we call the cleavage furrow. And that's an indication that the cytoplasm is about to be completely separated. Kind of think of it like tying a string around a balloon, where you get that cinching in the middle. The spindles are also starting to disappear, right? So those microtubules have gotten a whole lot shorter. Those are disappearing. Here's a side-by-side comparison of what we've seen so far minus prophase. So you can compare metaphase, anaphase, telophase, and cytokinesis, these three phases. But remember, this is not including prophase. So remember that P, M, A, T, prophase, metaphase, anaphase, and telophase. This picture is not showing you what's happened during prophase, only what's happening during metaphase and anaphase and telophase. So this is really good in that it shows you what the chromosomes look like at each phase. It also shows you that the cell is getting longer. Let's start with metaphase. Metaphase is one of the hallmarks of metaphase is that the chromosomes line up in the center, and that's really easy to see. In fact, meta means middle. And you've got those really long microtubules. And then if we move over to anaphase, the cell has gotten a little bit longer, which is characteristic of anaphase. But also now the chromosomes are starting to move away from one another. So think of the A in anaphase as away. The chromosomes are now separated and they're moving away from one another. In telophase, look what's happened. We've got those mitotic spindles are getting smaller. The nuclear envelope is reappearing. And also you've got that cleavage furrow. So this is essentially telling you, hey, these chromosomes have separated from one another. The cytoplasm has been divided, and now this, what was one cell, is just about to divide into two separate cells. I'm not sure why I have this same picture in here twice, so we'll just move past that. And then lastly, there's cytokinesis. And here now, in cytokinesis, the cytoplasm is completely divided into separate cells, meaning this cinching right here, or the pinching of what was one cell, is totally complete. And what you get are two separate cells, and we call those daughter cells. Okay, so now that we've gone through the entire cell cycle, remember there is interphase, and then there's the mitotic phase. That's the entire life of a cell. And the rate at which a cell goes through its whole cell cycle totally depends on the type of cell and some other factors as well. So let's take, for instance, the cells that line the inside of your stomach. They go through their cycle once every 24 hours. Your skin cells, for instance, take a whole lot longer than that. Cells in the kidney take longer than that. Nerve cells never undergo this once you're past the age of about 5. So cells in an organism's body totally divide at different rates. So this process that we've gone through here is going to vary depending on the type of cell. But some of the factors that control cell division are what we call, some of the factors would be, are there enough nutrients present for the cell to do this, to complete its life cycle? Another thing that is going to regulate cell division are what we call growth factors. These proteins that are going to stimulate cell division. Certain proteins need to be present in order for that cell to know to continue into the mitotic phase, to divide. Also, cells can't just go through this process unregulated. There have to be some processes that are going to tell cells when it's appropriate to divide and when it's not appropriate to divide. So there's something called density-dependent inhibition, and that's where when cells are crowded, they stop dividing. So when there are enough cells present in a particular tissue or organ, the cells actually go into kind of a resting phase. They're going to stop dividing, at least temporarily. And then another thing that's going to control cell division is what we call anchorage dependence. Cells need to be in contact with some other surface before they can divide. So your skin cells, for instance, when they're growing and they're going through their cell cycle, they have to be connected to another skin cell. If they're not, they won't undergo cell division. So there are growth factors that signal what we call the cell cycle control system. So these growth factors are going to be molecules that are going to trigger and coordinate events in the cell cycle. And through the cell cycle, there are checkpoints where if there's something wrong with the cell, or chromosomes haven't duplicated properly, or organelles haven't duplicated properly, those the cell cycle can be stopped. And essentially that cell is told, okay, you're not ready to divide. We're not going to allow you to continue through this cycle. Or if everything is okay and all the duplication of the cytoplasm and the chromosomes has been done correctly, then there are checkpoints in the cycle that can signal, okay, you can proceed to the next phase of the cell cycle. So let's look at those. There are 3 major checkpoints in the cell cycle. So first up, there's the G1 checkpoint. And this is a checkpoint that allows cells to enter the S phase. Remember, S stands for synthesis. And it allows the cell to go ahead and duplicate its chromosomes. If a cell isn't allowed into the S phase, the cell leaves the cycle completely and enters a non-dividing phase. Something's gone wrong, or there's a missing growth factor. Something has occurred not to allow that cell to progress in its life cycle. So then it doesn't go on to divide. It goes into kind of a resting phase that we call G0. There's G2, which is a checkpoint that allows the cell to go into the G2 phase of interphase. And then there's also an M checkpoint that allows the cell to go into mitosis. So there are checkpoints along the way. It's like quality control for inside of the cell. If everything is okay, then the cell is allowed to progress through these phases. If something has gone wrong in interphase, then these checkpoints allow for cells to stop what's going on and they enter a non-dividing phase. As you may know, there's a lot of research going on with regards to controlling of the cell cycle. Because when cells grow out of control, when cells escape or are allowed to proceed through these checkpoints, even when something's wrong, it causes the cell oftentimes to undergo uncontrolled mitosis. So that cell cycle and the checkpoints, that's controlling whether a cell can undergo mitosis or not. But when cells have something wrong, let's say a mutation in the DNA, when they have something wrong, sometimes they escape these checkpoints and are allowed to go into mitosis anyways. And this is essentially what cancer is. Cancer is uncontrolled cell growth. When cancer cells escape the controls in the cell cycle, the cells often divide very quickly, even in the absence of growth factors that should normally be telling the cell how to go through the cell cycle. And when those cells divide very rapidly, they they can often escape into your blood vessels. And once those cells that are mutated and growing faster than normal, once they get into the circulatory system, your blood vessels, then they have the ability to go anywhere in the body. And once they move, or once they have been transported to other areas of the body, they'll find a suitable place to start growing and dividing again. And when that happens, that's called metastasis. That's spreading of the cancer cells. So, for instance, my girlfriend who has metastatic colon cancer, the cells in her colon escaped this cell cycle, grew very rapidly, and were able to sneak into nearby blood vessels. And from there, those cancerous cells made their way to her liver, and now those cells are growing in her liver. And they're growing out of control, creating large tumors. A tumor is a cluster of cancerous cells that are growing very rapidly. From there, those cells left her liver. Some of those cells snuck out of her liver and made their way to her lungs. So now these cells are also growing and dividing out of control in her lungs, resulting in tumors in her lungs. So it's really important that we understand mitosis and some of the things that control mitosis, so that we can better understand how to stop and or treat these types of cancers. Okay, so we just left off on talking about a tumor. Remember, a tumor is essentially these abnormally growing cells that all cluster together. Not all tumors are bad. Benign tumors, they don't get into the bloodstream. They don't get into your blood vessels. They stay at the original site. And these are often very easily removed through surgery. Malignant tumors, those are the bad ones. Those are tumors where they've left the original location, like my friend's colon cells have left her colon, gotten into the blood vessels, and moved to other areas in the body. So that process of cancer spreading is called metastasis. Here's an image of, just for instance, breast cancer. So breast cancer is breast tissue is very well vascularized, meaning it has a really good blood supply. And it's got other vessels too that aren't carrying blood, but they carry kind of a clear fluid. These are called lymph vessels or lymphatic vessels. So if we look over at the first picture here, there's the tumor. That's that cluster of cells that are growing rapidly. And it may stay localized. At this point, it's benign. It hasn't spread. The cluster of cells is localized, just right there in that one spot. But what you'll see happen in the next picture is those cells are continuing to spread throughout the tissue. So there are a whole lot more cells here. And then because that tissue is very well vascularized with blood vessels and has lymphatic vessels, these cells, these purple cells here, have the ability, let me change my pen color. Oops. These cells now are going to leave the original site where they were growing, and they can sneak or slip right into those blood vessels, and those blood vessels can transport those cancers anywhere in the body. And so this is what we know as metastasis. Those cells also don't necessarily have to leave and sneak into blood vessels. They can sneak into lymphatic vessels as well. And those lymphatic vessels run all throughout your body. So these lymphatic vessels can carry tumors to other areas of the body too. So from here, I'm just going to let you read this last slide. And if you have any questions, please email me. Don't forget to watch the Amoeba Sisters tutorials. Also, go ahead and search any YouTube videos that might help you with this process. But keep in mind that your tutorial videos may include additional information that you guys don't need to know. So the Amoeba Sisters are pretty basic. I would watch the Amoeba Sisters videos, do their worksheets, and then augment that with the PowerPoints. Use those two resources first, and if you're still confused, reach out to me or find additional tutorials.